Okay, hello everyone. Welcome to the second day of PION's webinar entitled The Impact of Colloids and Nanoparticles on Intestinal Drug Absorption. That is part of our monthly fiber optic user group meeting series as well. I'm Sinko with PION, and I will be moderating this event today. We will have the last two presentations on this topic today, one by Dr. Patrick Sinko and one by Carl Box. We had a lot of questions yesterday, and I want to encourage you to contribute to a productive session today again by asking questions at any time during the presentation through the Q&A box on your screen. Any questions that we don't get to today will be answered by email after the event. If you need any technical assistance during the webinar, then please type your query into the chat box and send it to the moderator. Also, a recorded version of this webinar will be available on our website, and an email with the link will be sent out to the registrants as well. The first presentation by Dr. Patrick Sinko introduces the dissolution permeation model that describes the particle dissolution within the concentration boundary layer adjacent to a semi-permeable surface. Patrick Sinko is a researcher focused on reducing and replacing animals during preclinical drug development. He obtained his Bachelor of Sciences in Material Sciences and Engineering at Virginia Tech in 2013, his Master's in Material Science and Engineering at the University of Michigan in 2018, and his PhD in Pharmaceutical Sciences in 2019 at the University of Michigan. During his PhD, he developed a methodology to produce ultra-thin polymer membranes that could be used to study simultaneous dissolution permeation processes. After moving to the molecular pharmaceutics group at Uppsala University, he continues to work on designing a biorelevant benchtop device that will examine the dissolution absorption process of advanced drug delivery systems. Patrick, please share your screen and start the presentation.
Okay, thank you, Balint, for the nice introduction. Good morning, everyone. My name is Patrick Sinko, and I'm going to be talking to you about a model that we recently developed to describe the particle drift effect. And oops, there we go. So as we've been talking about through this series so far, the particle drift effect is a new type of particle dissolution model. Traditionally, as Dr. Zagano kindly introduced yesterday, we as the pharmaceutical community have assumed that particles don't necessarily interact with the concentration boundary layer, also known as the aqueous boundary layer or unstirred water layer. In fact, since there is no physical barrier at the so-called concentration boundary layer, particles can interact with the surface within that length scale, and I hope to at least show you some convincing evidence today that's true. So my talk will be going through a statement of the problem and showing the experimental setup that we use to test our hypotheses in our new model. I'll actually talk a little bit about the model and the predictions it's capable of making, and then get into the materials used to test this model. And finally, we'll cover the results of the dissolution absorption experiments performed in our new custom diffusion cell, and then end my talk with an interesting observation that we discovered during the model validation process. So based on some of the questions yesterday, I felt like it was necessary to add this slide and very briefly describe what a boundary layer actually is. So the formal definition of a boundary layer is described as a property reaching 99% of its far field value. So in the case of the particle dissolution problem, this is going to look at the velocity field and the concentration field. So if you can imagine the concentration very far away from a surface goes to zero. This is our classic bulk concentration equal to zero assumption. And I demonstrate this in these two plots from my most recent paper, where we plot the non-dimensional distance from the surface eta as a function of the value of the velocity on panel A function, and in panel B, concentration function. Now, these are the non-dimensional functions related to the velocity profile and the concentration. But when we say boundary layer, we're really talking about this point in space away from the surface where it's almost equal to this asymptotic value very far away. So the actual particle drift effect, it can be described like this, schematically at least, where we have some sort of solid membrane, semi-permeable membrane, biological membrane, and we have a fluid containing particles. We divide this scenario into three regions, region A, B, and C, where region A is our bulk region, that region of fluid that's very far away in terms of the velocity as well as the concentration at the surface of the membrane. Then B would be representing our concentration boundary layer, and we lump the membrane and the receiver compartment into region C. Take note that concentration boundary layers form not only at the surface of the membrane, but of course at the surface of particles as well. So to detect the particle drift phenomenon, the membrane should not be rate limiting in the mass transport. And things that can affect this can be, for example, the pH of your solution, which affects aqueous solubility, membrane diffusion coefficient, and partition coefficients. But as the concentration boundary layer is intrinsically linked to the hydrodynamic boundary layer or the velocity boundary layer, the hydrodynamics of your in vitro or in vivo model will affect this, this quantity. And of course, particles need to be small enough for this particle drifting effect to actually activate. The origin of the particle drift effect comes from a discontinuity of the flux. So traditionally, when we model this system, we say that the flux is equal in region A and region B across this interface, and then between B and C. But because of the presence of these new point sources of mass, we cannot actually say that the flux in A is equal to C, because we generate new flux because of those presence of particles in region B, or the concentration boundary layer. So how do we actually capture this on the bench? I was working with a device called the rotating membrane diffusion cell during my PhD, and semi-permeable membrane material called polydimethylsiloxane, or PDMS, which is a silicon rubber. So it's lightly lipophilic or organophilic. And from the data I previously generated, we saw that we couldn't achieve a membrane rate limited, or sorry, we saw that we were membrane rate limited for most of our measurements. So I designed a new diffusion cell, which was able to house a much thinner membrane, which allowed us to gain the maximum possible absorption rate for our model compound, ibuprofen. And we achieved this by using a technique called spin coating, where we take a large silicon wafer and deposit a water-soluble polymer like polyvinyl alcohol on the surface of this membrane. And then we know that PDMS is itself soluble in organic solvents, meaning that the solvents are orthogonal to each other between the pre-layer and the functional layer. What this allows us to do is then take that large wafer and actually deposit it into a petri dish full of water and allow water to infiltrate the radial edge of the membrane composite, releasing the functional material, the PDMS, which in this case is approximately 10 microns thick. And it self-assembles on the surface of the water, allowing us to extract it from the petri dish and install it in the diffusion cell. And you can see here the material is quite robust for being so thin. I'm actually poking it with tweezers at the surface of the water to demonstrate this in this middle panel. So now we have a diffusion cell, which we think it should be sensitive enough to actually measure any changes in the flux that is caused by particle dissolution within the boundary layer. So we use a very small volume of a small water column in this upper compartment to maximize the A to V ratio, getting our absorption rate as high as we can. And we use the spin coating technology to produce this 10 micron membrane. But are we really sure that what is the rate limiting process for our drug? So doing a quick calculation based on previous data, we saw that we needed to have a fully non-ionized ibuprofen to achieve the rate limiting process that is highest for the concentration boundary layer. So doing a couple of quick solution experiments, we see that in this using this new diffusion cell and membrane system, we achieve rapid steady state transport across the membrane with a very short lag time. And if I would had a pion rainbow analyzer, maybe I could have actually caught the little heel at the bottom, but unfortunately I was using HPLC at the time. And not going through the derivation of the math, but at least keeping in the mind the theory that we just talked about, this is what the ultimate flux through the membrane model looks like. And you can see here we have the diffusion coefficient for the drug, C0 being the solubility of the drug, and then a few repeated terms in this complex term at the back, where we have A, B, and lambda. And for the purposes of this talk, all you need to know is that A and lambda contain the particle dissolution model, and the B term contains the permeation or the partition model. Getting into what this model can predict, in panel A, we incorporate some more modern theories of mass transfer that were published recently by Dr. James Brasseur at Colorado Boulder and his then postdoc Yangshi Wang, I believe, who's now at Austin, Texas, and showing that we can achieve some enhancement due to the Sherwood number. We can talk about that more offline or in the questions session. The main thing I want to talk about in this slide is actually panel B, where we demonstrate the expected performance for ibuprofen as a function of the particle's radius. So as we decrease the radius, we see the expected increase in flux that we see in the particle drift phenomenon. But we also see that there's a dose dependence for this effect. In panels C and D, we compare the traditional film model for particle dissolution and then what our model predicts when we allow particles to dissolve in the boundary layer. In our traditional assumption, I model two theoretical particles, one with an average radius of about 77 microns and one with a very small radius, 1.2 microns. Each of these particles would be larger than and smaller than the respective concentration boundary layer. And you can see that the large particle sees a dose effect, increasing the flux through the membrane. However, the small particle, using the traditional model, basically achieves maximum flux at any dose because those particles are so small. When we allow particles to dissolve within the boundary layer, we actually predict that small particles can achieve a much higher flux than is possible when they are not allowed to dissolve in the boundary layer. So we hope to see this phenomenon in our results. To fully test the model, we took 3 particle fractions of ibuprofen. In the green, we have our high shear wet sieved material. This is the finest material that we tested, and it's our positive case where all of the particles in that sieve fraction exist as have a radius that's smaller than, or a diameter that's smaller than the actual concentration boundary layer that we're going to be measuring against. In the blue, we have dry sieve, very large particles, which serve as a pure negative case. In this case, all particles are larger than the length scale of the concentration boundary layer. And to challenge the model even further, we made a material that had both particles that exist below and above the concentration boundary layer length scale. So here we look at the receiver concentration profiles for the dissolution permeation experiments for ibuprofen in our new diffusion cell. And we can notice a few things. We have cell number 1 and cell number 2. I created two cells so that we could have at least some device effect involved here. And what you need to understand is that there's a slightly different hydrodynamic situation in cell 1 versus cell 2. So the concentration boundary layer, as measured by solution experiments, was actually about 10 microns thicker in the second diffusion cell. So we have a bit of ability to test how sensitive the model is. because of this difference in the two devices. The general trend is that smaller particles have a larger total permeated mass at the end of the experimental time frame, and also have steeper slopes. So this top row is our finest particles that are dissolving and permeating through the PDMS membrane. This is our intermediate or in-between particles, and then this is our largest particles. And of course, we see an increasing flux across as the dose number goes up, or the dimensionless amount of material that's in the system. So talking about our initial results, let's talk about the steady state flux that was derived from these dissolution permeation profiles. So I'm going to explain the plot a little bit here, and then we can jump into the results. So you're going to see this figure where I plot the log of the flux on the y-axis and a log of the dose number on the x-axis. At log of the dose number at equal to 0, we find a vertical line which indicates the solubility of ibuprofen. Any data to the left of this indicates that the material is in a solution state, and any data to the right indicates the suspension state for particle dissolution. In the solution side, we use the traditional film model to describe solution diffusion and permeation, and we can predict for diffusion cell 1 and 2 in maize and blue, respectively, for ibuprofen transport in that diffusion cell. In the right-hand side, in the suspension side, we have now 4 curves. The small dotted curves indicate the traditional film theory assumptions for particle dissolution, while the solid line is our new model. At the top of the graph, we have our maximum theoretical possible flux for this membrane drug system. As you can see here, when we take particles that all are smaller than the concentration boundary layer thickness, our model agrees quite well with the data, seeing a small difference due to the hydrodynamics at lower dose numbers, which then disappears as you increase the dose number approaching 1,000. Looking at our pure negative case, we again see a good agreement with the solution predictions and data generated from the diffusion cell. However, the data seems to be more aligned with the traditional film model assumptions as opposed to the under-prediction predicted using our model, which is good because we don't actually expect our model to be able to predict the particle dissolution in the boundary layer if the particles can't exist within the boundary layer. And the slightly higher fluxes here compared to the traditional film model are likely due to some settling effects, as we're working with a very shallow water column, and even at high agitation speeds, settling of these large particles likely occurred. So maybe the more interesting result between these two is these in-between particles, where we have particles that exist above and below the concentration boundary layer thickness. It appears, using the mass average radius, that at lower dose numbers, the particles seem to behave more towards the traditional assumptions in the film theory model, while at higher relatively higher dose numbers, the data seems to deviate completely. So we did a bit of thinking about why this might be. And we went back to the basics of what happens during the particle drift effect, which is that small particles infiltrate the concentration boundary layer, or at least the distance from the surface, which is the concentration boundary layer. And if we assume good mixing, where particles are relatively evenly distributed, that means that we have many more small particles that are likely existing within the boundary layer and influencing the flux that is produced across the membrane. Also, smaller particles in a steady state context have a higher dissolution rate than their larger counterparts because of their larger surface area to volume ratio. So when we looked at the number average radius for this distribution, we saw that our predictions lined up beautifully with the observed data, indicating that, at least during steady state processes, the smaller particle radii likely dominate the dissolution process and also the permeation process. And again, the way we argue this is that in the mass or volume fraction, most of our particles by mass or volume actually are above these vertical dotted lines, indicating that they're larger than the concentration boundary layer. But when you look at the distributions as a number fraction for both of them, you'll see that a significant and maybe even the majority of particles actually are these microparticles able to be dissolved in the concentration boundary layer. And finally, I wanted to talk about a bit of an interesting observation that occurred during the development of this diffusion cell to test this model. So the final diffusion cell used this laser cut support system where we took stainless steel sheets and cut out a grid pattern and sandwiched our very thin membrane between the two to create a very sturdy support for this very thin polymer membrane. However, when we started, we didn't have access to this laser cutting technology, and we actually used sonic sieve meshes to support the membrane material. So taking two of these sonic sieve meshes, cutting them and shaping them to be an appropriate fit for the diffusion cell, and then mounting it inside the glass chamber that you saw earlier in the presentation. The issue with this is that these woven wires actually create pockets that are almost a millimeter deep as compared to the very flat planar surface of the custom laser support. What this allowed was for particles of all sizes to get trapped in these deep wells where we had very nice flow of the fluid and particles over this flat surface in the final diffusion cell. And what we saw was when particles were getting trapped in these wells at the surface with the same concentration boundary layer thickness, we saw that the particles actually responded with a particle drift-like effect, where we see this increase in the measured flux, but we lose the dependency on the particle radius. Again, because we're basically pinning those particles to the surface and creating those point sources of mass at the surface, you get this enhanced flux. But because they're not moving, they're not convecting with the solution, then you lose the size dependency. So we actually saw this and said, wow, this is probably really what's happening with the human body when we have the microvilli and villi structures and those deep crypts that exist between those two biological structures, where the body is able to concentrate particles in those crypts to enhance nutrient and pharmaceutical absorption. So with that brief overview of that work, I want to leave just 3 points with you. First, that small enough particles can dissolve within the concentration boundary layer, which is not a physical boundary layer, but just a point in space where the gradient becomes very, very shallow for the property of interest, whether it's velocity, heat, temperature, or concentration. And when these particles move within the length scale of the concentration boundary layer, they enhance mass transfer across the membrane because they act as discontinuities in the flux profile. During pseudo-steady state phase, mixtures that contain particles that are larger than and smaller than the concentration boundary layer can initiate this particle drift effect until those smaller particles are all dissolved or become a less dominant part of the dissolution process. Again, we saw this when we looked at number fraction versus mass fraction for this in these suspension experiments. And highly textured surfaces, particularly in vitro, can trap particles near an absorbing surface, inducing an effect that's similar to the particle drift effect, but it's not truly the particle drift effect. And this is also likely a mechanism that our biology has taken advantage of for hundreds of thousands of years. So with that, I will thank my funding sources for my PhD. That was AbbVie Pharmaceuticals and the FDA. And of course, my thesis committee, the Amadons, Dr. Sun and Robert Ziff in physics at the University of Michigan. I also had a lot of support in lab from fellow graduate students at the time, Nilafar Salehi, Nicholas Job, Ravi Sharma, and Jill Wenderot, and my students at the time, Troy Halstead, Sarah Harris, and Yue Yuan. And of course, support staff at Michigan, including Xiaomei Chen from the David Smith Research Group and the U of M 3D printing lab, where we had done some related work to this, and the tech transfer office there. And I guess this is a bit old of a slide because I actually have acknowledgements for Sirius Analytical for Paul and Carl and Haley, and then the Pion team at the time, which was Michael Kelly, Konstantin Sinman, Okansa Sinman, and Dave Kawajski. And with that, I would enjoy taking any questions. Thank you.
Thank you. Thank you very much, Patrick. I actually have a question to start with, more on the device that you have mentioned. Can you tell us a little bit on the volume and the surface area and the unstable layer thickness? What's the range where it can be varied in that system, and have you utilized that in these studies? Does it make sense for your in vitro studies to play with the unstable layer thickness?
Yes. So the device itself, when we were operating for these experiments, was about 15 milliliters of fluid. So in the 100 millimeter, or sorry, a 75 millimeter diameter upper chamber, we filled it just above the shaft of the impeller that we were using, which was a kind of a modified hydrofoil. And the reason we did this was hydrofoils are known to be good at resuspending particles. And the model that we were able to obtain was shallow enough so that we could push down the volume of the donor phase to increase our total absorption rate, because absorption rate is a function of that A to V ratio. And the impeller itself was probably, I think, about 30% of the diameter or 40% of the diameter, which gave us enough room to insert probes and things on the side of the device to measure pH or to take samples conveniently. And in this case, we had the protective stainless shield mesh on top and bottom of the membrane. So we were actually pretty safe when we were poking either pipette tips in or installing the hydrofoil impeller into the device, because the stainless steel obviously is very tough and protected our very thin membrane. This is not fully optimized. I think where we left it was that the hydrofoil impeller itself could be further designed to take up more of the internal diameter. That way we get even better particle resuspension. But for based on the data we obtained, at least so far, the stir system seems to be adequate.
Thank you. We have a few more questions. The particle drift effect helps to explain deviations from theory. Is there any consequence in vivo?
Yes, like Dr. Zagano said yesterday, if particles are able to drift into, well, the concentration boundary layer and then ultimately the mucus layer, they can get trapped within the crypts of the villi and microvilli, where they are going to be very close to that absorbing surface. And because of that proximity, the concentration will be that's evolved from the surface, gets rapidly absorbed by the epithelial layer, which leads to increased absorption in the biological system.
Thank you. Thank you. Another question, how can we analytically distinguish the drifting particles input into permeation if the solubility of the drug is unknown?
Yeah, that's tricky because every particle dissolution model that I know of at least requires knowledge of the solubility in one fashion or another. So I would suspect that you would have to have a measurement for that or an estimate of that. And like Dr. Lee was talking about yesterday with these amorphous solid dispersions, finding solubilities in amorphous forms or non-thermodynamically stable forms could be quite tricky.
Thank you. One last question. Have you tried to visualize the particle drifting effect, and is it possible?
I would say in our device, it would not be possible to visualize it due to the fact that working with large pieces of curved glass and very dense suspensions, and where we have to point the microscope would be in 30 or so microns, 40 or so microns above the surface. Actually, that's what I forgot to answer from Balan's question. The concentration boundary layer thickness in this experiment for diffusion cell 1 and 2 is 30 and 40 microns respectively. So it's quite tricky in a large scale device like this. However, using something like a microfluidic channel, likely you could visualize this, but it would be a much more specialized setup than this more generic dissolution device that we operated with.
Yes, thank you. And thank you very much again for the great presentation.